Hello all. Today's class will be discussing microbial nutrition. As we all know, nutrients are very important for all of us and microorganisms for growth. Nutrients help in biosynthesis, that is construction of new cellular components, and energy production for day-to-day growth and metabolism. Nutrient requirements are as important as environmental factors, and based on the amount available or amount required, we have macronutrients and micronutrients. Macronutrients, as the name suggests, are required in larger quantities by microorganisms. Carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium, iron are the macronutrients, and they make up over 95% cell dry weight. Carbon, oxygen, hydrogen, nitrogen, sulfur and phosphorus are components of carbohydrates, lipids, proteins and nucleic acids. The others, potassium, calcium, magnesium, iron, they are present in the cell as cations with a variety of roles. For example, potassium K+ is required for the activity of many enzymes, including those involved in protein synthesis. Calcium Ca2+ is important in heat resistance of bacterial endospores and has many other functions related to enzyme activities. Magnesium Mg2+ is a cofactor of many enzymes, complexes with ATP, and stabilizes ribosomes and cell membranes. Iron as Fe2+ and Fe3+ form a part of cytochromes and is a cofactor for enzymes and electron carrying proteins. So those were the macronutrients. Now we have micronutrients or trace elements: manganese, zinc, cobalt, molybdenum, nickel, copper. They are needed by most cells but only in very minute quantities. They form part of enzymes and cofactors and act as catalysts for reactions and have roles in maintenance of protein structure. The interesting part about them is that they are present as contaminants in glassware, water and regular media components. So we need not add them as such into the media, and it is very difficult to demonstrate a micronutrient requirement of an organism. That is, since they are present as contaminants in water, glassware, etc., there is never a lack of micronutrients, and it is difficult to demonstrate that growth is dependent on them because there is never a lack of it. We cannot just demonstrate that is required for growth. So in nature, micronutrients are ubiquitous and do not usually limit growth. Among micronutrients, zinc is present at the active site of some enzymes and is involved in the association of subunits of enzymes. Zinc Zn2+, manganese Mn2+ aids many enzyme activities, again catalyzes the transfer of phosphate groups. Molybdenum MO2+ is required for nitrogen fixation. Cobalt CO2+ is a component of vitamin B12. There are some special requirements for particular microorganisms depending on their morphology or the environment which they are in. For example, diatoms require silicic acid to construct the beautiful cell walls of silica. Another group of microorganisms, halophiles, they require large amounts of sodium or potassium. They grow in saline lakes and oceans with high concentrations of sodium ion. So these are examples of special requirements other than macro and micronutrients. Whatever be the nutrients, microorganisms require a balanced mixture of these nutrients. If an essential element is short in supply, microbial growth will be limited regardless of the concentrations of other nutrients. So they should have everything in balanced mixture, and not that they have more of one thing and less of another thing. That will affect the growth. Now coming to the major ones, carbon, hydrogen and oxygen. Carbon is needed for the skeleton or the backbone of almost all organic molecules that we know of. Carbohydrates, proteins, lipids, everywhere carbon is the backbone. And usually molecules which are carbon sources also contribute oxygen and hydrogen atoms. Thus, they form the source of all three: carbon, hydrogen, oxygen. They come together. The requirements for carbon, hydrogen, and oxygen are thus satisfied together by different carbon sources. Carbon sources also serve as energy sources since they are almost always reduced and have electrons that they can donate to other molecules. Because of this, they are the building blocks, and they also serve as energy sources because they are reduced and they have electrons which can be donated to other molecules. The more reduced the organic molecules are, higher their energy content. Example, lipids have higher energy content than carbohydrate. This is because electron transfers release energy when the electrons move from reduced donors with more negative reduction potentials to oxidized electron acceptors with more positive potentials. So electron transfers from a reduced donor to an oxidized acceptor that releases energy. So more reduced organic molecules are, higher the energy content. Now carbon dioxide is an example of a carbon source that is oxidized and lacks hydrogen, so does not supply hydrogen or energy. Reduction of carbon dioxide is a very energy expensive process, and many microorganisms cannot use it as a sole source of carbon. They use more reduced complex molecules such as glucose, which are already preformed, as carbon sources. So carbon dioxide, very few organisms use it as a sole source of carbon. Most of them would like to go for reduced complex molecules such as glucose as carbon source. Now when it comes to carbon sources, microorganisms utilize all naturally occurring carbon sources. Bacteria, Burkholderia cepacia can use over 100 different carbon compounds, but some bacteria are fastidious. They use only some carbon compounds. They require specific compounds. Some like methyllotrophic bacteria metabolize methane, methanol, carbon monoxide, formic acid and related one-carbon molecules. Leptospira, which is a pathogen, use only long-chain fatty acids as the source of carbon and energy. So these are examples of fastidious bacteria, specific requirements. Now actinomycetes, another group of microorganisms, they can degrade amide alcohol, paraffin and even rubber. So almost all carbon compounds are utilized by different microorganisms. In natural environments, we even find complex populations of microorganisms degrading or metabolizing indigestible human-made substances also, such as pesticides. We call them xenobiotics. Even that can be degraded by microorganisms. But usually, different populations work together to degrade such compounds. And they are degraded in the presence of a growth-promoting nutrient that is metabolized at the same time. This process is called co-metabolism. So they will be feeding on and digesting on a growth-promoting nutrient, and in that process, the other compound, which is usually an indigestible human-made substance, that also gets degraded. So this process is called co-metabolism. Advantage of this process is that products of this breakdown process can be used as nutrients by other microorganisms, which normally cannot feed on this indigestible substances. So here we see a give and take policy of the microorganisms, whereby indigestible substances are made accessible to another group of microorganisms by co-metabolism. So that was about carbon. Next we have nitrogen. Nitrogen, as we all know, needed for the synthesis of amino acids, purines, pyrimidines, carbohydrates, lipids, enzyme cofactors, etc. Nitrogen can be incorporated as ammonia directly through the action of enzymes like glutamate dehydrogenase or glutamate synthetase. So microorganisms can incorporate ammonia by the action of these enzymes. Ammonia is converted to amines, which is then incorporated into amino acids. Assimilatory nitrate reduction is seen with most phototrophs and many non-photosynthetic microorganisms, whereby they reduce nitrate to ammonia and incorporate the ammonia. So nitrate reduction, nitrate is reduced to ammonia and this ammonia is incorporated. This is assimilatory nitrate reduction. Assimilatory means they assimilate the compound produced by this process. So nitrate reduced to ammonia and ammonia is assimilated in assimilatory nitrate reduction. This is seen in case of most phototrophs and many non-photosynthetic microorganisms. Nitrogen is also incorporated by nitrogen fixation, which is performed by cyanobacteria and symbiotic bacterium Rhizobium, where they reduce and assimilate atmospheric nitrogen using a special enzyme system called nitrogenase system. Here, nitrogenase enzyme fixes atmospheric nitrogen, makes it available in other forms such as ammonia, which can be readily utilized by plants and these bacteria. So nitrogen is seen in symbiotic bacterium Rhizobium and many cyanobacteria also have it. Cyanobacteria can be both free living and symbiotic. Rhizobium is usually performing nitrogen fixation in symbiotic association with legumes. Next, phosphorus. Phosphorus is present in nucleic acids, phospholipids, nucleotides like ATP, several cofactors, some proteins, and other cell components. Low phosphate levels limit microbial growth in many aquatic environments, so it is important there. All microorganisms use inorganic phosphate as the phosphorus source and can incorporate it directly. The uptake by E. coli has been studied. Bacterium can use both organic and inorganic phosphates. Organophosphates such as hexo-6-phosphates can be taken up directly by transport proteins like porins. Other organophosphates are hydrolyzed in the periplasm, which is seen in the gram-negative bacteria, by the enzyme alkaline phosphatase to produce inorganic phosphate. Inorganic phosphate is transported across the plasma membrane by specific transport systems. Phosphate uptake by E. coli has been studied. This bacterium can use both organic and inorganic phosphate. Organophosphates such as hexo-6-phosphates are taken up directly by transport proteins. Other organophosphates are hydrolyzed in the periplasm by the enzyme alkaline phosphatase, and inorganic phosphates are released. This is then transported across the plasma membrane using porins or other phosphate-specific transport systems. Sulfur is needed for the synthesis of amino acids like cysteine, methionine, some carbohydrates, biotin, thiamine, etc. Most microorganisms use sulfate as a source of sulfur and reduce it by assimilatory sulfate reduction. Sulfate is reduced and the compounds formed are assimilated by the microorganisms. A few require ready-made available reduced form of sulfur such as cysteine, that may be fastidious microorganisms which require the reduced form of sulfur as cysteine. Now, microorganisms in general grow and reproduce when minerals and sources of energy, carbon, nitrogen, phosphorus and sulfur are supplied. They have the enzymes and pathways necessary to synthesize all other components required. But some microorganisms lack some enzymes. They cannot manufacture all these compounds, so they need certain substances or their precursors from the environment as growth factors. So growth factors should be supplied for some microorganisms readily in the environment or as the precursors to facilitate growth. Organic compounds required because they are essential cell components or precursors of such components and cannot be synthesized by the organism are thus called growth factors. Three major classes of growth factors are there: amino acids, purines, pyrimidines and vitamins. Amino acids, as we all know, are needed for protein synthesis, purines, pyrimidines for nucleic acid synthesis. Vitamins are small organic molecules that usually make up all or part of enzyme cofactors and only very small amounts sustain growth. So they are required in very small amounts, vitamins. But some microorganisms require many vitamins like Enterococcus faecalis, which needs 8 different vitamins for growth. Other than vitamins, other growth factors are also there, like heme, which is required by Haemophilus influenzae. Heme comes from hemoglobin or from cytochromes. Some mycoplasmas need cholesterol. Now, this knowledge of specific growth factor requirements, which microorganism requires which growth factor, that can be the basis of microbiological assays to quantitate these growth factors. For example, Lactobacillus and Streptococcus species are used in microbiological assays of most vitamins and amino acids, because these organisms have a requirement of for those amino acids and vitamins. They cannot produce them by themselves, so they will grow only if they are incorporated in the medium in which these organisms grow. So depending on their growth, we can understand how much of these growth factors are present in that media. So that forms the basis of microbiological assays. Just to explain, appropriate bacterium is grown in a series of culture vessels, each containing medium with an excess amount of all required components except the growth factor to be assayed. A different amount of growth factor is added to each vessel. The growth factor quantity or concentration is then plotted against the extent of bacterial growth. Ideally, the amount of growth resulting is directly proportional to the quantity of growth factor present. How much our growth factor is present, that much increase in growth is observed. We can say that there is a correlation between the both. Microbiological assays are specific, sensitive and simple. Common assays are that of vitamin B12, biotin, etc. Now, in a test sample, the quantity of growth factor is determined by comparing the extent of growth caused by the unknown sample with that resulting from standards. So, standard curve is plotted taking the concentration of growth factor corresponding to the growth of the bacteria. From that curve, we can find out the amount of growth factor produced by a test sample, an unknown sample. So this is how microbiological assays are performed. Now, because microorganisms are found to synthesize vitamins, they have been employed in industrial level fermentations also. Some common examples would be production of riboflavin by species of Clostridium candida, Ashbia, Erythrothecium, etc. Production of coenzyme A by Brevibacterium, production of vitamin B12 by Streptomyces propionibacterium pseudomonas, production of vitamin C by Gluconobacter erwinia cornybacterium, production of carotenes by Dunaliella, vitamin D by Saccharomyces, etc. are some common examples of industrial fermentations. So that was a brief account of different nutrients required by microorganisms and what are they required for. In addition to these nutrients, carbon, hydrogen, oxygen, microorganisms also require energy source and electrons for growth. Based on the sources of carbon, energy and electrons, we have different nutritional classes of microorganisms. First, we start with carbon. Based on the source of carbon, microorganisms can be divided into autotrophs and heterotrophs. Simple definition: autotrophs can use carbon dioxide as the sole or principal source of carbon. Many microorganisms are autotrophic. They carry out photosynthesis and they use light as the energy source. Some autotrophs oxidize inorganic molecules and derive energy from electron transfers, and those are the chemoautotrophs. If they derive energy from light, they become photoautotrophs. If they derive energy from electron transfers, they become chemoautotrophs. The second group is heterotroph, the organisms which use reduced preformed organic molecules as carbon sources. These preformed molecules normally come from other microorganisms or other organisms. Most heterotrophs use reduced organic compounds such as glucose as sources of both carbon and energy. So based on carbon, we have autotrophs and heterotrophs. Now, based on sources of energy, which we discussed just now, there are basically two sources of energy, light energy and energy derived from oxidizing organic or inorganic molecules. Phototrophs use light as their energy source. Chemotrophs obtain energy from the oxidation of chemical compounds, either organic or inorganic. Now, based on the source of electrons, they are classified as lithotrophs or rock eaters they are called. They use reduced inorganic substances as their electron source. And organotrophs extract electrons from organic compounds. So, if it is inorganic substances, lithotroph; organic substances, organotrophs, whichever serves as source of electrons. Based on carbon sources, we have autotrophs and heterotrophs. Autotrophs use carbon dioxide as a sole or principal carbon source. Heterotrophs rely on reduced, preformed organic molecules obtained from other organisms. Based on energy source, phototrophs rely on light energy. Chemotrophs rely on oxidation of organic or inorganic compounds to obtain energy. Based on electron source, lithotrophs or rock eaters, they use reduced inorganic molecules, and organotrophs, they obtain electrons from organic molecules. Based on the primary sources of carbon, energy and electrons, microorganisms fall in 4 nutritional classes. They are photolithotrophic autotrophs or photolithoautotrophs, photoorganotrophic heterotrophs or photoorganoheterotrophs, chemolithotrophic autotrophs or chemolithoautotrophs, chemoorganotrophic heterotrophs or chemoorganoheterotrophs. As the names suggest, we find photolithotrophic autotrophy or autotrophs have light as a source of energy, water or inorganic compounds as source of electrons, carbon dioxide as source of carbon. So, photo, light; litho, rock eaters, chemicals or inorganic compounds as source of electrons; and autotrophy, autotrophs, carbon dioxide as the carbon source. Examples: algae, cyanobacteria, purple and green sulfur bacteria. Algae and cyanobacteria can employ water as electron source, whereas purple and green sulfur bacteria need inorganic compounds as the electron source. Next, photoorganotrophic heterotrophy. These organisms, again as the name suggests, has light as the energy source, organic compounds as the electron source and carbon source. Purple non-sulfur bacteria and green non-sulfur bacteria are the common examples. Now, chemolithotrophic autotrophy or chemolithotrophic autotrophs, going by the name, they have inorganic compounds or chemicals as the energy source and the source of electrons. Since they are autotrophs, they use carbon dioxide as a source of carbon. Common examples: sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria, iron oxidizing bacteria. The last class, chemoorganotrophic heterotrophs or chemoorganotrophic heterotrophy. These organisms, again going by the name, chemoorganotrophic heterotrophy. So, they have organic compounds, organic chemical compounds as the source of energy, electrons and carbon. Common examples: protozoa, fungi and most pathogens or non-photosynthetic bacteria. So, these are the different nutritional types of microorganisms, and among these 4, most of the microorganisms fall in 2 groups: photolithotrophic autotrophs, which are often called photoautotrophs or photolithoautotrophs. They use light energy and have carbon dioxide as the carbon source, as we mentioned. Eukaryotic algae and cyanobacteria employ water as electron donor, releasing oxygen. Purple and green sulfur bacteria cannot oxidize water but extract electrons from inorganic donors like hydrogen, hydrogen sulfide, elemental sulfur, etc. So, either water or inorganic compounds are used as source of electrons. The second group which most microorganisms fall into is the chemoorganotrophic heterotrophs, or called chemoheterotrophs, chemoorganoheterotrophs, or even just heterotrophs. They use organic compounds as sources of energy, hydrogen, electrons, and carbon. Frequently, the same organic compound will satisfy all these requirements. All pathogenic microorganisms are chemoheterotrophs. So, as mentioned, large majority of microorganisms so far studied are either photolithoautotrophs or chemoorganoheterotrophs. The other two nutritional classes have fewer microorganisms, but they are also very important ecologically. Photoorganotrophic heterotrophs or photoorganoheterotrophs, they are common inhabitants of polluted lakes and streams. Some purple and green bacteria are the examples. They are photosynthetic and use organic matter as the electron donor and carbon source. Some of these bacteria can also grow as photoautotrophs instead of being photoheterotrophs. Chemolithotrophic autotrophs or chemolithautotrophs oxidizes reduced inorganic compounds such as iron, nitrogen, or sulfur molecules to derive both energy and electrons for biosynthesis. So they depend on inorganic compounds for energy and electrons. Carbon source is carbon dioxide since they are the autotrophs, but they can also derive carbon from organic sources and then they become heterotrophic. So chemolithotrophs can exist both as autotrophs and heterotrophs. They contribute greatly to the chemical transformations of elements in the nutrient recycling that continually occur in the ecosystem, conversion of ammonia to nitrate, sulfur to sulfate. So chemolithautotrophs have a major role in that. Now, just like what we mentioned now, a particular species usually belongs in one nutritional class, but some show metabolic flexibility and alter their metabolic patterns in response to environmental changes. For example, many purple non-sulfur bacteria, they are photoorganoheterotrophs in the absence of oxygen, but at normal oxygen levels, they oxidize organic molecules and function chemotrophically. So usually they are photoorganotrophic heterotrophs, but sometimes they become chemoorganotrophic heterotrophs also. And when oxygen is low, photosynthesis and oxidative metabolism may function together simultaneously. So in the absence of oxygen, photosynthesis occurs. Normal oxygen, chemotrophy they resort to, and at low oxygen level both can function simultaneously. And some other bacteria such as Bigetova, they use inorganic energy source or organic carbon source or even sometimes carbon dioxide. So such microbes are called mixotrophic because they combine different metabolic processes. This sort of flexibility may appear complex and confusing, but for the microorganisms it gives a definite advantage if the environmental conditions go on changing. So they are mixotrophic to survive in the environment, in the changing environment. So here we end the microbial nutrition, different nutrients required for the growth of microorganisms and different classes of microorganisms based on their mode of nutrition. Thank you.